brought to you by Curious Brandland. Here's the deal. What we need to understand about biotechnology requires a lot of vocabulary. So we're going to look at this handout, and what I want to do is show you the basic arrangement of the handout and tell you what you're going to do as a class exercise. Question? No. All right. So first, did you guys count? Make sure you have 15 vocabulary words. Good, good. Right? We're going to use these words and create a little evaluation. You're going to make a quiz, and then you're going to take someone else's quiz. But let's talk more about the handout, and I'll finish with the quiz, and we'll stop recording. So you got 3 parts. You got a list of vocabulary words that you'll see in anything biotechnology. Right? What is that they use it to say biotechnology? It's exploded over the last couple of years with the Human Genome Project and different techniques. I'll elaborate on some of the things that we do now that graduate students did earlier. Right? There's certain things we have high school students doing. That's amazing. If we could afford to do all the labs. So you got your vocabulary, then you got your concepts. So you're going to see something. Some people need to focus right now. All right? Concepts. So this is transgenic organism. The steps to making it. So let's follow through. Then it tells you, goes into more steps about using DNA as a vector, inserting DNA. So there's a whole bunch of things on this note packet that I'm giving you. Gel electrophoresis. Okay, that's a big one. Something called PCR, polymerase chain reaction, in the Human Genome Project. This part of the packet is how to do it. So what's next? Application of technology. So the way this packet goes, see later on, I'm going to give you some review questions for your real quiz. And one of the questions says is, well, what are some transgenic organisms? Well, here it is. Earlier in the packet, it said how to make a transgenic organism. Now they're saying, what's the application? Like animals, plants, bacteria. I really want to talk about bacteria in a second. The Human Genome Project. So this is application. So I had a student last year like, this is listed twice. My response is because it's listed twice. One is listed to tell you how to do it. The other one's telling you how we use it. There's a subtle difference. So, really today you're going to look at the vocabulary list, and then here's one, gel electrophoresis again, but how it's used as a separation technique to determine paternity tests. So we're gonna look at this next class for do the whole baby daddy thing. You are the father. We'll figure it out. Okay. All right. So without all the yelling and screaming and bleeping out of things, okay? So, and then here's something. What do we use this technology for? Not just paternity tests, but DNA samplings, maybe in a crime scene, identifying unknown victims or perpetrators. And then the last thing, mitochondrial. I should say that better. Mitochondrial DNA. Mitochondrial DNA. Earlier in the year, we learned about mitochondrial and chloroplasts having their own DNA. There's an evolutionary relationship there, and we've actually been able to track maternal DNA. The other way they describe the mitochondrial, they call it maternal DNA. This is different than the DNA in your nucleus that you inherit from both parents. Right? Egg and sperm fuse together. The sperm cell doesn't have much cytoplasm, but the cytoplasm and the organelles you get from your mother. So we've been able to track this and track evolutionary relationships and divergence over years by looking at the mitochondrial DNA. It's another way of looking at comparisons of species as well. So let's look at your this whole transgenic thing, and then we're going to talk about the quiz, and that's it for me talking today. All right, so transgenic. We use bacteria. So how many have heard of bovine, the phrase bovine? What's it mean?
Oh, related to cows.
Cows. Okay, so to use the example of diabetics, being that I am one, although I don't have to inject insulin, in the old days we used bovine insulin, which means we'd extract the insulin from the pancreas of a cow. We're not the same species. It saved lives, but it wasn't as effective as human insulin. So now, when a person needs an injection of insulin, they inject something that was mass-produced by a bacteria. Doesn't that seem like a contradiction? I just said bovine wasn't as good, but now we're using something produced by bacteria. However, it's human insulin. This technique was created before you guys were even born. So this is actually more of a history lesson. So once we were able to purify this insulin and get, if you inject insulin from another human, you're better at. And now with designer gene therapy, they can take your own gene, stick it in a bacteria, and it's going to mass produce it. So, quick refresher: bacteria, are they prokaryotes or eukaryotes? Prokaryotes. What are we? Eukaryotes. We're eukaryotes. That's the miraculous thing about it is we take eukaryotic genes and we stick it in a prokaryote. One of your list of vocabulary words, we can go back up here, make sure you get this. All right? Here's some big ones. All right, recombinant DNA, transgenic. A transgenic organism is an organism that has DNA from another organism. Bacteria have this unique ability to take in foreign DNA and express those genes. What does it mean to say express genes? We've taken 2 tests where we had to know this.
Shows the traits.
It shows the traits. What do you mean?
Like the physical characteristics of that trait.
The physical characteristics of that trait. Well, how do you get there from the DNA to the
genotype? Like expression of a gene is like what trait shows that is coded for by the DNA.
Coded for. But how do you get there? The DNA is a nucleus.
Oh, translation.
Translation, what happens before translation?
Transcription.
Transcription, translation. That stuff we learned a couple chapters ago is not going away. Okay? Because we'll talk about evolution. We'll talk about replication errors. How that can affect the ultimate expression of genes. Maybe it's a mutation, and then we have a new variation of the trait. So, bacteria through something else. I told you to look up a picture earlier called restriction enzymes. The unique thing is restriction enzymes can cut out a segment of DNA. So this is a loop of genetic material that's a bacteria. We cut a piece out, stick a new piece in. That new piece is, say, the insulin gene, and that bacteria shells it out by the gallons. We've been able to mass produce it because these prokaryotes are expressing eukaryotic genes. AB Bio, some of you may take that your junior or senior year. I was explaining to someone, like I was flying and plane, I was telling them what some of our students are doing. They were amazed because what we have 11th and 12th graders doing, they're actually genetically engineering a bacteria. We take the gene for resistance to ampicillin, which is an antibiotic. We stick it in this bacteria. We grow it. We put some antibiotic in their growth media. We call it the agar, or the growth media. And we put the bacteria in there. If they took the gene in and expressed, they survive. It's called a negative and positive control. Like if they don't take in the gene, they don't survive. So we look to see those colonies. They have actually expressed a gene for resistance for an antibiotic. And we got 11 and 12th graders. That same technique, when I was in college, you had to be like a junior, senior, or even a graduate student to be able to do it. So there's the technology is amazing. We can't afford to do all those labs, so we save some of those for AB Bio. So there's another term here, the word plasmid. The interesting thing about bacteria, they got their main genetic material, and they have these small circular loops, and those loops are how we get them to take in foreign DNA. I had a professor in college who said, bacteria are interesting in that they're like, he called them nucleotide hogs. Like if you put genetic material in their media, they're gonna swallow it up. They're gonna take it in through them, like diffusion, endocytosis. They're gonna take in that genetic material and express those genes. And fortunately for us, they can do that, and they take any type of genetic material and they can express it. We've benefited from that. All right. So what we're gonna spend most of class doing is we're gonna make a quiz. You have 15 lists of vocabulary. You have the term and its description. Term and a description. So, how are you going to do it?
Matching.
Okay, matching is probably a good way. But I have one requirement. I do not want one monster thing. I don't want 1 through 15 matching that way. I want you to break it into parts. You can make the quiz any format you want. Okay? Any format you want. So, say you want to break it up, say you want to do like 1 through 8 and make that your part 1, and 9 through 15 that your part 2, you can do it like that. Or 1 through 7 and 8 through, I don't care. So you can make my only requirement is that you have at least 2 sections. Right? And then, look at it like this. That could be your column A, and then the vocabulary description, there's the word, the name of the process could be your part B. Make it A and a B column if you want. If you want to make it multiple choice, I don't care. So that's one version. Alright, I've had some students do it like this, where they just, because there's 15, they just separate and make that part 1, make that their part 2, and this their, so they just do every 5, they make that into a section. Alright, this is what, you're making a quiz, you're going to have to make a quiz, a separate sheet for your answer key, then you're going to fast it forward, and you're going to take someone else's quiz. It will be your responsibility. Whoever gets your quiz, they're going to give it back to you. You will grade it. Alright? And I will collect the actual ones you make in your key, but the ones that you guys take, you'll get that back yourself. Are there any questions? Yes.
Is this the biotech quiz we have to
Yes,
do?
you're making a biotech quiz, and then you're taking another student's quiz. Now, this actually should be homework for next class period. The actual quiz I'm going to give you is next Monday.
So we're not allowed to use notes or anything on the quiz we're taking now, or are we?
Okay, you're going to use this packet to make a quiz
Just on the vocab?
on the 15 vocabulary. And you're going to take someone else's quiz as if it were a quiz.
Okay.
And then you're going to grade whoever took yours. You can discuss it however you want. What is my only requirement?
Broken up into parts.
You have at least 2 parts. You can do multiple choice. You can do matching. You can do fill in the blank. Remember, someone in this class is going to take your quiz, so if you want to win friends and influence people, go ahead and make it as difficult as possible, or make the bare bones. See if they know what they're talking about. That's a good practice. Okay? I think we're pretty much done.